We are live now.
Yeah, thank you, Peter. Hello, everyone. So before our talk begins, let me give a brief introduction of Dr. Mansi Mungee. So she's currently a research fellow at the University of Leeds, UK. Her domain expertise spans wide across community ecology, macroecology, biodiversity conservation, and policy making. She's particularly interested in understanding spatio-temporal pattern in the diversity and assembly of ecological communities. For her PhD research at IISER Pune, she looked at the patterns in diversity and community assembly of two different two different animal groups, hawk moths and birds. Our PhD research result lead to the development of a novel method for obtaining morphological traits of nocturnal insects from digital images. She's currently working on a project called Drivers and Repercussions of UK Insect Declines, or DRUID for short. This large collaborative project focuses on collating and synthesizing all available datasets on insect declines across UK and determines spatio-temporal pattern using multiple indices such as richness, biomass, abundance, etc. So, and today she will be talking about the case study on macroecology. So now over to you, Mansi.
Right. Thank you, Yambin. That was quite a good introduction. Sometimes it's difficult to remember everything that you've done, so it's nice to be introduced sometimes as a reminder for yourself. Right. So, hi everyone. The introductions are already done, so I will just dive into the case study. As mentioned, the case study that I'm presenting was actually a part of my PhD work, which was about 4 years ago. And that study was comparative study between birds and hawk moths. And today is all about birds, so I will focus on just that part of my PhD. I will share the screen. Right. Can you all see this?
Yes, we can see the screen.
Okay, great. Excellent. Right. So I will be talking about using trait and phylogenetic diversity to evaluate the generality of stress dominance hypothesis in communities of East Himalayan birds. Lots of big scary words in there. Don't worry, we'll break it down. We'll take it one by one. I will first start with a brief overview of what I mean by communities and community assembly, and then what I mean by trait and phylogenetic diversity. And finally, how I'm going to use these community assembly processes with trait and phylogenetic diversity to evaluate something known as a stress dominance hypothesis. So let's start with the basics. Community assembly, functional and phylogenetic diversity. What are these terms? What do they mean? Why are they important? Why am I talking about them today? And before I begin, I just want to sort of emphasize that these are really vast, mutually non-exclusive subjects within community ecology. And it is quite impossible to cover all three in great depths in this one lecture. I believe there is some additional reading that you guys were shared or will be shared that you can go through. But today I will be covering only the aspects of these topics which are relevant for understanding stress dominance hypothesis. So what is community assembly? Very simply, the word assembly in English means a collection of or putting together of similar objects or objects with a common purpose. In the context of ecology, community assembly refers to the processes which determine what species will be found in a given location. So we are talking about the processes which have put a collection of species together. Why are these species found in South India and not in North India, for instance? What has led to the formation and maintenance of this particular community? So without any background also, with just intuition and just common sense, I think we can all come up with an exhaustive list of these processes. Why are these species found here and not there? Is it because of the environment or the abiotic factors that these species like, which is why they are found here? Is it because of the presence or absence of another species, such as a competitor or a symbiote, which we can call biotic factors? Or is it just a completely random chance dispersal event? This species came here because of illegal wildlife trade, for instance. The species actually had no role to play. It was because of a random process. So these are all what we refer to or call as community assembly processes, the processes which are responsible for determining what species will be found where. Now, in these three examples that I just talked about, biotic, abiotic, and stochastic or randomness, these are broad categories, and within them also there are many subcategories that I will not be talking about. But very at a general level, the biotic and abiotic factors are what we call deterministic or niche-based processes. And these are the processes that I will be focusing on more. In my experience, it really helps to understand these processes better with the help of a schematic that I've used here. And I will be sticking to this schematic throughout this talk to explain different processes. So let's try and understand this as much as we can in the very beginning. So here, the schematic that you can see on the right. I hope, can you see my cursor? I hope you can. So this is a hypothetical mountain system, right? And across this mountain, there are many different species found. Let's take the example of birds. There are many different birds which are found across this entire mountain range. The sum total of all the birds that are found across this mountain constitute what we call a regional species pool, which is your outermost panel. So these all different species represented in different colors and shapes are what we call a regional species pool. Now, from this regional species pool, there are certain species which are found at a particular location along this elevational gradient or this mountain system. And different local communities will be found at different elevations, right? This is also very straightforward. As you go up a mountain, you see different kinds of communities. You see alpine species at the higher elevations, and lowland species are very different, right? So let's take the example of a mid-elevation community, which is a nested subset of your larger regional species pool. Now, the community assembly processes that I'm talking about should help you determine how this local community is subset or formed given a regional species pool. So at the first level, let us consider abiotic filtering or environmental filtering. So only the species which possess the traits or which possess the capacity to survive in the environment of a given elevation will be able to survive or persist in that elevation, right? So from the regional species pool, the alpine species will be subset and filtered at higher elevations, others at lower, and similarly at different elevations. So let's say the environmental filtering determined this particular set of species. They all have very similar traits, very similar ecologies that can help them to exploit the resources and survive the environment at the middle elevation. So that was your environmental filtering. Now, because environmental filtering has selected the species which are all ecologically very similar, because they have the traits to survive at a narrow given elevational band, they will also compete more strongly, right? Because they're all very similar to each other. So to reduce that competition, the next filter or the next assembly process, which is your interspecific competition, that will eliminate certain species and minimize that competition. So these two, which were very closely related, are now farther from each other because the central species are removed by the second filter. And what you get is a net local community, which is a trade-off between environmental filtering and interspecific competition. Or in very simple terms, this net local community is a trade-off between how similar the species can be, right? Because if they are too similar, they will not be able to coexist. They will just compete each other outcompete each other. So it's a trade-off between how similar and dissimilar the species within a local community can be. Now, this is all nice and good for a theoretical framework, for a conceptual framework, but how do you actually go about testing some of these things in nature, right? How do you test for the presence of environmental filtering or interspecific competition? And this is where the concepts of functional diversity and phylogenetic diversity come in the picture. So to understand functional diversity, we must first understand what are functional traits. So the definition says that functional traits are morphological, physiological, or behavioral characteristics of individual organisms relevant to their environmental response and or their effects on the ecosystem. That's the definition. But to understand, very simply, now instead of asking why are these birds found here, I am asking why are these beak sizes found here. So I have used a trait, which is the beak size, as a surrogate for species, right? And this automatically makes the entire framework a little more measurable or quantifiable. I can quantify beak sizes, beak lengths, beak depth, beak width. And the information on beak size automatically tells me about the relationship between the bird and its environment, right? So because of the beak size, I know the kind of resources that particular species will exploit. I automatically know how different or distinct a hornbill is from a warbler, for instance, right? Because their beak sizes are so different. Right. So now if I just translate the previous schematic of community assembly processes along elevational gradient, instead of species, I now translate or surrogate or substitute the species by traits. The same schematic will look very similar, except the terminologies will change. So now I have a regional trait pool instead of a regional species pool. The environmental filtering will select for very similar traits, that is very similar beak sizes in the example that I'm using. And all these beak sizes allow species to exploit the resources found here. Because competition will not allow very similar beak sizes to coexist, it will eliminate certain beak sizes and create gaps. And finally, you will have a local community, which is trade-off between very similar and very dissimilar beak sizes, right? So now immediately we have a testable, quantifiable framework to assess the presence of these two filters. So if in my local community, the beak sizes are very similar as compared to a random subset from the regional trait pool, then I know that environmental filtering is dominant in that community, or the assembly process which dominates the assembly of that community is environmental filtering. But if the beak sizes are very dissimilar from each other as compared to a random subset, then we know that interspecific competition also plays an important role. Now, there are certain right. So before I proceed, I just want to emphasize that this framework of using functional traits to study community assembly processes this has become one of the most important analytical benchmarks in modern community ecology. And there are a lot of different applications and a lot of different processes that you can study by compiling very good trait data for your field organism, for your study organism, or your organism of interest. Now, this framework, of course, has its own limitations. The first is that in the previous example, we talked about beak sizes, which means that we are assuming that the environment also acts on beak size and that the beak size also plays a role in competition. This may not always happen in nature. We know beak sizes are involved in competition between species, but are they also important from environment point of view? How does temperature select beak sizes, or how does precipitation select beak sizes, right? These are all aspects of the environment. So we need to be very careful, and we need to be very, we need to back the choices of our traits by some ecology. we need to ensure that the traits that we have chosen are involved in competition as well as involved in survival or tolerance of the abiotic conditions. Secondly, most traits are often correlated, right? For instance, beak size actually scales with body size. So if you have a larger body-sized bird, it will also have a larger beak size. So how do you then determine whether the assembly process is acting on beak or is it acting on body, but just getting translated to beak because the two traits are correlated, right? So these are some of the aspects that one needs to be careful about. Finally, it's almost impossible to measure all ecologically relevant traits of the species, right? Because there are many traits, like the definition mentioned, there are morphological traits which are more straightforward to measure, but there are also behavioral traits, right? Songbirds have different display behaviors. There are different physiological traits such as thermotolerance or habitat use or foraging specializations. So these are important traits, but they are slightly difficult to quantify. And even if you can quantify a few of them, it's almost impossible to measure every single trait of every single species in your study system. And this is where the concept of phylogenetic diversity comes in the picture. Right, so what is phylogenetic diversity? So we talked about species diversity, we talked about functional diversity or trait diversity, and the third most important dimension of biodiversity, which is incredibly increasingly used as a surrogate or a complementary metric to quantify the diversity of the region, is phylogenetic diversity. Very simply, it represents the diversity of lineages in a community. So for instance, if you have two communities, right, and both of them have 10 species and very similar beak sizes, we know that the species richness of community A is equal to species richness of community B because they have both 10 species. So species richness is identical. We know that they have very similar beak sizes. So their functional diversities are also very similar. But now what if the 10 species in the first community actually come from the same genus, and the 10 species in the second community come from 10 distinct genera? So this means that the phylogenetic diversity is actually much higher in the second community than the first community, even though they have similar species richness and similar functional diversity. And this simple distinction can actually be very useful from a conservation point of view in certain scenarios where you're trying to conserve maximum diversity, because distinct lineages will have distinct evolutionary histories and therefore will present a richer system. So this is the third surrogate of diversity. Now, the simple rationale for using phylogenetic diversity as a surrogate for traits is that, first let us just go back and discuss this schematic from the point of view of functional diversity first. So instead of a regional species pool and a regional trait pool, we have a regional phylogenetic pool that you can see here. The environmental filtering will select for similar species because it is mostly true that closely related species have similar traits. We know two species of hornbills will be more similar to each other than a hornbill and a warbler or a magpie, right? So closely related species will be subset by your first assembly process, which is your environmental filtering. Then interspecific competition will remove closely related species which are too similar to each other to give you a net local community. Now, the basis for this entire schematic is, of course, that closely related species are more similar to each other. And this is something that is known as phylogenetic niche conservatism, which means that the niches or the requirements of two species are very similar to each other when they are closely related on a phylogenetic tree. So this entire schematic can actually be very useful to study how community assembly processes change along an elevational gradient. And stress dominance hypothesis actually makes a prediction of how these assembly processes will change. Will environmental filtering dominate at higher elevations, or will its strength be higher at the lower elevations? So when integrating all these concepts together in just one schematic, if you see here, so in the topmost panel of this schematic is a phylogenetic tree, which represents 11 species that constitute your regional species pool, right? So the regional species pool consists of 11 species in 3 distinct clades, that is 3 different, we can call them groups or genera or families within the larger regional pool. So the species within a clade are more closely related to each other than species across a clade. Traits are conserved, so which means that species within a clade also have similar traits to each other than traits across a clade. Now, under strong environmental filtering, the species with it, so in the bottom panel are these 3 hypothetical local communities, which are constructed or assembled under different conditions or different assembly processes. Now, in community A, when strong, under the conditions of strong environmental filtering, the species within a local community will exhibit functional and phylogenetic convergence or clustering or under dispersion, which means that the traits in these species, in this green, in this first community, they will all be very similar to each other. And because traits are conserved, these species are also going to be very closely related to each other. So under strong environmental filtering, you will have convergence of trait and phylogenies. In this community C, under conditions of strong competitive interactions, you will see trait and phylogenetic divergence, which means your species are now coming from 3 distinct clades. That is, they are as far from each other ecologically as possible because competition was really strong in that community. And then you have a scenario in which you want to compare whether your filtering and competition are strong or no, and which is why you construct something known as a null community or a random community, which is basically simply saying that you write a computer program which takes random subsets from this regional pool to create a random community, which does not assume the presence of clustering or divergence. So then you can compare your natural community to the random community to comment on how the natural community is assembled. So this is the entire framework that actually talks about how community assembly processes can be studied using functional diversity and phylogenetic diversity. And the patterns in functional and phylogenetic diversity, that is whether the patterns are clustered or over dispersed, can help you to understand this relative strength of these 2 different assembly processes. Now the question arises, how do these processes change along an elevational gradient, or do they actually change along the elevational gradient? And this is where the stress dominance hypothesis comes along. So the stress dominance hypothesis makes an important and explicit prediction that the strength of environmental filters should increase towards the highest elevations because the abiotic conditions are stressful. And those stressful abiotic conditions will select for species with very specific adaptations that should be able to survive in such conditions. Whereas in the more benign or stable conditions of the lowest elevations, because conditions are benign, stable, populations flourish, and competition becomes more important. Specialization becomes more important. And because the external pressures are minimized, competitive interactions are maximized. So we can again look at this in the form of a schematic, and the same schematic as before. So you have under harsh or stressful environments, you have a clustered community or a constrained community, which is very similar to each other, the species, and are all equally adapted to the harsh environment. Whereas under benign or stable environments, competitive interactions are maximized. So along an elevational gradient, because higher elevations are stressful because of thermoregulatory constraints, air density, resources, the environmental filtering dominates, and at lower elevations, you have competitive interactions dominating. So if I have to test something like this along an elevational gradient, I will just measure or collect data on ecological communities along a gradient. I will measure the traits and phylogenetic distances within each community, and I will just calculate some metric of divergence or convergence. Basically, I will just calculate what are the distances, pairwise distances between all co-occurring species, and I will plot those distances along elevation. And if stress dominance hypothesis is true, it actually occurs in nature. If my hypothesis is true, then what I will see is that this metric of divergence reduces with elevation, which means I find over dispersion at lower elevations, which is this, and I find clustering or low divergence at higher elevations, which is this. All right. So this was stress dominance hypothesis. This is how it is studied. This is how you go about testing something like this, using functional and phylogenetic diversities. And this is exactly what I did in an extension to my PhD work using communities of birds. So I will quickly talk a little bit about the primary field data that was collected. Right. So this study was conducted in Eagle Nest Wildlife Sanctuary. I'm not sure, but perhaps some of you have heard of this. This is nested within the larger Eastern Himalayan Global Biodiversity Hotspot, so which is this region. And you can see in these panels, these two panels, your top panel is actually the total threatened species richness in the world. And this is just the species richness across the world for birds. And the Eastern Himalayas are high on both of these panels. So the study site is actually located within this larger region. It's a global biodiversity hotspot of immense conservation value. And it's actually it has the highest density of passerines anywhere in the world, right? So within this larger region, Eagle Nest is actually a very small sanctuary in Arunachal Pradesh. It's located here in the western Arunachal Pradesh, bordering Bhutan and Assam on the south. So Eagle Nest is actually a very small protected area of just about 280 square kilometers, right? But it is truly remarkable because in this small region, the elevation changes very steeply from 500 meters along the southern border, which is here, to almost 3,000 meters along the Eagle Nest Ridge, which is somewhere here, right? So the straight line distance between these two highest and lowest elevation points is actually just 20 kilometers. But the temperature difference between them is more than 20 degrees. So to see this temperature difference, you will actually have to travel 2,000 kilometers of latitude. But here you see that temperature difference in just 20 kilometers. So this presents actually a remarkably steep elevational gradient. So, yeah, so this actually presents a remarkably steep elevational gradient to study how community assembly processes will change in a relatively feasible, practical, or manageable spatial scales, right? Secondly, the bird diversity in this region is spectacularly high. So you actually get a very high diversity within local communities. And that is why you can assume that competitive interactions will play an important role in these bird communities. So you have a strong environmental gradient to study environmental filtering. You have high diversity of bird communities to study competitive interactions. And you have a stress gradient because you have an elevational gradient. So you can assume that stress is higher at higher elevations. And which is why this entire system presents an ideal ecological laboratory to study stress dominance hypothesis in nature. Right. So, right, before I go any further, my PhD was on birds and hawk moths, like I mentioned. The hawk moth part was actually my sort of baby, and I have been more inclined towards studying insects in general. And the bird part of my data was collected by this remarkable guy, Rohan Pandit, here in the center. Rohan is a phenomenal bird watcher. He was a project student in the lab back then. And he conducted these truly impressive transit counts for birds and gave me a data set of almost 15,000 or 16,000 individual birds along 2,600 meters of elevation, right? Representing 245 species and 50 families. It's a remarkable data set. So if I look at just simply species diversity with elevation, I can see that my diversity peaks somewhere at around 1,200 meters along the elevational gradient. And there's low diversity at the lowest and highest elevations. Now going ahead and checking stress dominance hypothesis in these communities of birds. So we need 3 raw ingredients to study stress dominance hypothesis. The first, of course, is your primary data, which is your species data, which means you need to know what species are found at which elevation, which is what Rohan gave me. The second thing I need is functional traits for these birds. So which I compiled based on previous works in the region and from secondary data or literature, because birds are one of the better studied animal groups out there. You have a lot of information on their traits. You have global trait databases from which you can extract the traits of your species of interest or the species in your data. So I compiled these different traits, 8 to be precise: wing length, tarsus length, body mass, beak size, primary substrate, foraging height, primary diet, and habitat. Now, all these traits actually have different ecological functions in birds and are associated with different ecological axes in birds. And this is some of the information that is available from previous studies, the role of a trait in a bird's ecology. So for instance, wing length affects dispersal ability of a bird. Tarsus length affects foraging ability of a bird. Body mass affects both the thermoregulation and the amount of resources that a bird will need. So these 3 traits can be overall considered to play, these are morphological traits which can be expected to play a more dominant role in stress tolerance. Because when resources are limited, body size will be affected. When air density decreases with elevation, dispersal ability via wing length will be affected. And similarly, as habitats change with elevation, tarsus length becomes increasingly more important or less important. So the effect of environment on these 3 traits is expected to be more pronounced than the other remaining 5 traits, which are your beak size, which is more involved in competition. Again, primary substrate is where a bird obtains its food from. Species with similar substrate behaviors will be expected to compete more strongly. Species with similar diet will be expected to compete more strongly for resources, right? So these are all known as alpha traits, which are involved in competition, and beta traits, which are involved in stress tolerance or abiotic tolerance. So we have the primary data, which is where species are found, which species are found where. We have the traits, which is your functional data. And then we have the phylogenetic data, which is also available for the birds of the world. And it's readily available. There's a freely available resource for birds. So you can just download a phylogenetic tree for the species found in your data set and calculate the distances between species via their branch lengths. I will, of course, not go into the details of this. But the most important thing to remember is that we assumed that closely related species will have similar traits for this framework to work. So you can actually test for some of these things. For instance, here on this phylogenetic tree, I have colored the branch lengths by the body size, right? So the body size actually goes from red to blue, from small to large bodies. And you can see phylogenetic niche conservatism. Just visually, you can see that the colors tend to be conserved across the phylogeny. So you have this entire clade, which is more similar in body size. You have this clade here, blue, this clade. So in general, there is a propensity or a tendency for closely related species to be more similar. But of course, this is just visually. There are statistical tests that you can do to quantify some of these things, right? So you establish that connection first before going forward, that your traits are actually conserved across the phylogeny. And I will not go into the details of that. Right. So now you have the 3 raw ingredients. That is, you have your species data, you have your functional trait data, and you have your phylogenetic data. Now what you need is a metric of divergence or a metric of convergence. So for that, I used these 2 different metrics known as mean pairwise distance and mean nearest taxon distance. I will again not go into the details of this, but basically what you're doing is you're calculating the distance or the difference between the traits of all co-occurring species, right? So if you have 10 species occurring together, A to J, I calculate the distance between the traits or the difference in the trait value for A and B, A and C, A and D, right? So all pairwise distances, and I take the mean of that. So that's my mean pairwise distance. And a similar metric is mean nearest taxon distance. Now I will just plot this distances across my elevational gradient, across the elevation, and I will expect to see something like a decrease in divergence. And because I want to see whether the patterns are actually different from a random community, I will also simulate or generate random communities from the data, which is known as null assembly. You can do that using a computer program, but and there are many different ways in which you can do this. I don't want to go into that, but what you're trying to see is whether the patterns that you find actually are significantly different from a random community or no, right? Because you can expect, this is a very hypothetical extreme case scenario where you will see all species from a green clade. In nature, in empirical communities, it will be slightly more complex. You will probably see 2 green and 2 red, right? And in that case, you don't know whether it is random or competition or filtering. So you actually generate a random community and then you check your observed community against a random community. So that's typically how you do any assembly process investigation. So this is the expectation. I expect my metric of choice, which is MPD or MNTD, and I expect both of these metrics for both of my indices, functional and phylogenetic, to decrease with elevation. So I did this for my birds, and let us see what we found. Right, so this is what I found. In general, you can see that there is a larger tendency of a negative pattern. That is, my divergence is reducing with elevation, which means that higher elevation communities are actually more clustered or more under-dispersed. That is, environmental filtering dominates at higher elevations, right? So the first row is actually the functional alpha traits, which are the traits involved in competition. The second row is my functional beta traits, that is the traits involved in environmental filtering. And the third is my phylogenetic diversity. So in general, for all 3 kinds of metrics and across all different metrics of divergence, there is a larger tendency for communities to be more dissimilar within each other, the species in a community to be more dissimilar from each other at lower elevations and more similar to each other at higher elevations. Except in these 2 cases, observed MNTD and SES MNTD. So this is my observed values, and this is values as compared to a random community or a null model. So to understand why this happened in case of these 2 metrics, I actually calculated the same index that I've used here for each trait individually. So here I have used alpha traits as all the alpha traits together and beta as all beta traits together, which means I've taken body size, wing length, and tarsus length together. And for the alpha traits, I have taken the habitat, foraging, beak, and diet together. So when I look at the individual traits individually, so this is my wing, tarsus, body, which is my beta trait, and my beak, habitat, foraging, substrate, and diet as my alpha traits, I see that there are differences which are important in terms of the strength of the process acting on that trait. So this means that different traits are actually involved in a particular assembly process to varying degrees. And this is true for any ecological community anywhere on the globe, right? The strength of the community assembly process will be very context dependent. You will not get the same effect of environment on the beaks of Himalayan birds as you will get on the Amazonian birds, right? They will play different roles. They will play similar roles, but to different magnitudes, right? The effect of environment is dependent on the environment itself. And because the environment changes, the effect will also change. So different traits actually give you different information about the assembly processes. And this is something very important to be noted. So you see very strong environmental filtering happening for body and tarsus and wing, but very weak effects on the foraging mode and almost no effect on primary substrate. This is also sort of expected because I mentioned that this is a very steep environmental gradient, right? Environment changes really rapidly. So you do expect the effect of environment to be pronounced here in this case. So overall, what this study showed us was that first, the stress dominance hypothesis actually makes important predictions for how community assembly processes change along tropical elevational gradient. And those predictions are at least valid for Eastern Himalayan birds of eagle nest, right? In general, this prediction has been found to be valid for a lot of other places and not just elevational gradients. Any stress gradient that you can think of. It can be an anthropogenic disturbance gradient. You don't really have to go to the Himalayas to study something like this, right? It can be like a rural to urban gradient. It can be a gradient of pollution. It can be a temporal gradient of successional stages of a forest, right? After disturbance. It can be a pH gradient along a shoreline. So any stress gradient where the gradient, environmental gradient varies from benign to more stressful for any particular organism of choice. Finally, functional and phylogenetic metrics of diversity, they provide important and complementary information to test assembly processes in natural empirical communities. Because without functional and phylogenetic diversity, we just have species present along a gradient. We don't know how these species are related to each other. So to quantify the species relationships, whether they are more related to each other or less related to each other, you need empirical metrics of functional and phylogenetic diversities, which can translate your species relationships to quantifiable metrics and also ecologically relevant metrics. It is important to ensure that the traits that you're using are conserved across the phylogeny because you want closely related species to be more similar to each other. Because if they are not and convergent evolution is happening, then you can get a convergent pattern, but because of competition, right? Convergence is due to environment, but you can get it because of competition if there is convergent evolution. Finally, different traits may be involved in different processes, and the strength of the process will change depending on the trait, depending on the taxon, and depending on the region. So it is important to not only look at multivariate diversity indices, like combining all traits together, but also look at individual traits that can inform the process that you're studying. And yeah, this was it, more or less. I have no idea how I did time-wise. Right.
Yeah, hi. I think we have two questions in the chat box. I think we can, you can address it.
Sure. Yeah. So I'm trying to maximize this thing again. I just can't. Okay, fine. Yeah.
Yeah. Okay.
I can't read the questions in the chat, I think.
Yeah, I will read it out for you. Is it okay?
Yeah, great.
Yeah. So the first question is from Sachin, and he wants to know what are the factors affecting species to compete with each other and not compete for a given habitat? Like, say, for instance, adapting for the resource availability,
Right.
etc. Like,
Right. So intuitively, you can imagine that species which will compete with each other will have similar requirements, right? So if you have two species and they are both insectivores and they both coexist, then they're automatically competing for resources, right? So if you're talking about factors which affect competition, then I would say it is the species ecology itself, their requirements, how much their niche overlaps with each other. And it is, of course, the environment also. Some species compete more in a given environment. When the resources are limited, for instance, competition will be more fierce. And the population sizes, right? So if they are both have high population sizes, competition will be more fierce.
Yeah,
Just to
I
add on-
hope.
Just to add on to that, because
Yeah.
we see that I agree to the fact that if they have similar requirement, there'll be competitions. But MacArthur's Wobbler's case was the opposite case, right? Where they tried to segregate or partition the resource to get the maximum benefits, and maybe they did not compete, right?
So, yeah. So what you're talking about is actually speciation and species diverging to avoid competition, right? So what I'm talking about is not really on evolutionary timescales. You see, you go to a forest, you see a community, and you try to find the processes that have put these species together right now. I'm not talking about what has happened in the past. What you see today, you try to deduce the processes responsible for that.
Okay, so you mean
Yeah.
to say that probably those species might have competed earlier,
Correct.
the past, and but then later on.
Yeah. So this is a snapshot study.
Right.
Yeah.
Thank you.
Yeah, the next, I think the next question is from Govind Sudarshan, and he wants to know how is random assembly formed?
Right. So this is actually sort of a philosophical debate, because if an assembly cannot be attributed to the process that we are studying, we call it random, right? So if I can't find environmental filtering or competition, I will call it random. But in general, randomness is the absence of a process. So one example I gave was because of dispersal because of illegal trade, right? So a species was actually transported from where it was supposed to be by a trader to another location. It just happened that the environment of that location is also suitable for that species. It escaped and it survived. This is a random event. Another random event can be chance dispersal because of a storm. A small bird was just transported from very far from its location because of high winds. This is another random process. So these are all, basically these are chance events that have nothing to do with the species ecology itself, right? So it is not related to the species abiotic requirements or a species ability to compete with its neighbors. So it's not the place where the species evolved, but it's the place where it is found now because of other things.
Yeah. The next, there is another question from Govind, and he is asking, how does stress hypothesis affect migratory species? Does the environment, I'm not sure what he's trying to say, does the environment for competition affect them more? I hope like the question I'm reading out is correct, Govind. I think you can unmute yourself and then ask the questions directly.
I can't hear you, Govind. Have you?
Yes, can you? Am I audible now?
Yes, yes, yes.
Yes. My question was, in terms of migratory birds, is there in terms of size of the population, does the more environmental filtering, or does the competition factor affect them more?
I think his voice is breaking. So the first question is, how does stress hypothesis affect migratory species? I think we can address that. And for the remaining, I think Govind, you can ask your questions in a discussion forum. For now, I think the network might be the network problem.
Yeah.
So we can address them in the discussion forum.
Yeah.
So you can address for this, how does stress hypothesis affect migratory species? So I think you can address this now, Mansi.
Right. So Govind, we couldn't hear you properly, so I will just address the question that was in the chat. How stress dominance hypothesis affects migratory species, right? So now, if you recall, in one of the earlier slides, I talked about how stress dominance hypothesis does not really affect anything, right? It's actually just a prediction. So you are testing whether that prediction is true for different communities of birds. Now, when you look at migratory species, in my data also, along an elevational gradient, when I study different, or when I count different birds, some of those species are migratory. So it depends on, because my transects were done in the breeding season, how are the communities that are observed in the breeding season assembled? That's the question, right? Now, when I do the same community again in the wintering stages or in the wintering season, then I will be able to comment on whether the same prediction comes true, which means whether the species that are now found in the wintering stages along this elevational gradient, whether they are also dominated by environmental filtering at higher elevations and benign conditions and competitive interactions in the benign conditions, right? So because stress dominance hypothesis makes a prediction for an environmental gradient, so you are not predicting anything for a single species or a single community. Your prediction is for a series or a network of communities along an environmental gradient. You can assume that the prediction will actually remain true because migratory species will be equal in number more or less at the different communities, right? But whether it's actually true in nature or no is something that one needs to check. And we have data for wintering. It's a paper that I'm right now working on. So if you actually send me an email in a few months, I will be able to answer that. But I don't have the answer right now.
Yeah, the next question, I think we'll take up this question, and the remaining questions we can address in the discussion forum. So today's last question is from Lakshmi Bharadwaj, and he's asking, how does stress hypothesis affect or fit in a mixed species flock community? This is our first question. So, and the next question is, what is the one major factor or trait apart from bill length which has produced most notable results in your studies? Does this hypothesis fit for those species which are affected by anthropogenic disturbances?
Right. So I'll answer the first one, and then I think you'll have to repeat the second one again.
Yeah, sure.
So the first one is about how the stress dominance hypothesis fits in a mixed species flock. Right?
Yes.
So again, this is similar to my previous answer. So you will need mixed species flocks at different ends of a gradient, and then you can ask the same question. But in general, I would expect that for mixed species flocks also, the effect of environment is going to be different when a mixed species flock is found at lower elevation versus when it is found at a higher elevation. So they definitely do flock together for certain common requirements. So they are definitely, they have similar ecologies, but whether they are more similar or less similar by chance is something that needs to be addressed along a gradient. So behaviorally, you are basically reducing the subset to a very specialized group now. You're not looking at all passerines or all birds. You're looking at just very specific mixed species flocks. So because this is again a subset already, so you already have an environmental filtering constraint here, right? Because the constraint is that these species are similar to each other in their behavior. They all flock together. So I would expect them to have similar prediction as SDH, but of course the magnitude will actually be much less than what you observe for a whole species assemblage, all bird together. And for your second question, I will.
Yeah, the next question is like, what is the one major factor or trait apart from bill length which has produced most notable results in your studies?
Body size. But the problem with body size is it is involved in so many life history axis, right? We know body size is involved in thermoregulation. We know body size is involved in resource competition because larger bodies means more resources. We know body size scales, or many traits scale with body size, allometric dependence. So it's actually, the problem is not whether a trait is showing the expected pattern or no. The problem is that you're not really sure what the trait is involved in for your study system, right? You actually have to do manipulative experiments or physiological experiments to correctly assess the role of body size. But because it's such a multivariate trait, so is beak size actually, and which is one of the main points I'm making in my paper, that these traits are not really, it's not very straightforward to link them to a process, right? It's easier to do it in plants where these experiments have been performed. We know the exact function that leaf surface area has, or that the width of the stem has. We don't know the exact function of these different traits for other animals. We know they are involved in things, but that's the problem. They are involved in things, plural, not just one. So it's a little complicated.
I think she has already explained for the continuation of the same questions. And she also wants to know whether this hypothesis of the correlation between bill length and the for the production of the community assembly. So whether these are also affected by the anthropogenic disturbance, like the hypothesis fit for those species which are affected by the anthropogenic disturbance.
Yeah. So actually what I forgot to mention is that when you take traits which are correlated, such as, wing size, beak size, and tarsus length scales with body size, you actually take residual wing size or relative wing size, which is that you take the wing which is excess after the relationship with body is removed. It's again a method of reducing correlations among traits. That is something that you do. And for your anthropogenic disturbance, it's a valid disturbance gradient. And stress dominance hypothesis has actually been studied along anthropogenic gradients. So you need a continuous gradient of low to high disturbance. If you can quantify disturbance in such way, in some way, or if you know that the disturbance gradient is true, then you can sample communities of birds along that gradient and test for the stress dominance hypothesis.
Yeah, I think we will end the session with these questions, and further questions, everyone, you can put in the discussion forum, and order if it is okay with you, Mansi, I think they can directly address the questions to your mail, ask questions if
my email address has been shared with them, or I can just give it now if people are interested, or you can just share it with them later. I think they should have it.
If anyone wants to directly this thing, you can always put in the discussion forum, and I'll be troubling you if there is questions in the discussion forum.
Sure, no problem. Yeah.
So,
cool.
Yeah. Thank you very much, Mansi.
No problem. Bye, everyone.
Bye. Thank you, everyone.
Thank you. Bye.